So for purposes of the final exam, we're going to take a look at viruses. We normally only spend a very short time in General Bio 1 on this, but considering the current state of affairs, I thought it would be useful to put a little more emphasis on viruses and viral infection in general. So this is page 107 of the lecture outline. Got a couple of quick notes here. First of all, what is a virus? Viruses are not cellular. If you think about the beginning of the semester when we talked about what a living thing is, one of the things we said was that living things are all made of cells. Well, viruses are not made of cells. Viruses, you might see them defined as being obligate intracellular parasites. So let's break down that term, obligate intracellular parasites. Obligate means that they have to work this way, they can't work a different way. They're obliged to be intracellular parasites. What's an intracellular parasite? Well, intra means within, inside of. So they have to parasitize cells by getting into those cells. They're not going to be able to use a cell as a host unless they cross that cell's membrane and get inside and sort of hijack the inner workings of the cell. And a parasite, you're probably familiar in general with what a parasite is. It's some biological entity. It may be another animal, it may be a bacterium, it may be a fungus. In this case, it's a virus that uses a living thing as a host and sort of steals things from the host, steals nutrients, steals machinery, borrows some capacity that that living thing has in order to carry on some of its own activities. So, in other words, to do what they do to host organisms, viruses have to get inside the cells of the host organisms and parasitize them, do something beneficial to them and detrimental to the host. They're obligate. They can't work any other way. In other words, viruses by themselves, sitting on this piece of paper, sitting on a table, sitting on the surface of your body, do absolutely nothing to you. They have to get inside your cells to do something bad to you. So what are viruses if they're not living cells, or if they're not made of living cells? I thought I'd just put a couple of terms out here, and some of these terms are also up here. Some features of a virus. Viruses don't have cell membranes because they're not cells. But really what they are is just a little bit of genetic material surrounded by a protein shell. And I'm going to post a video that describes what I'm about to say, so please watch this video and then watch the next one. The protein shell that the other video refers to is what's correctly called a capsid. So viruses have a capsid. It's just a little protein shell, a protein coat that it sort of houses the virus. And I won't even attempt to draw this. The video will. I don't draw very well, so you'll get more benefit from looking at the other video that I post. So the viral capsid is this little shell that holds what's inside. Well, what's inside the virus? Genetic material. That could be DNA or it could be RNA. Now, genetics is a topic that you cover in detail in Bio 106, so without going into a tremendous amount of detail here, let's say that in a typical cell, you're going to have both DNA and RNA. They're both types of nucleic acids. If you think back to when we did organic molecules earlier in the semester, we mentioned these, didn't go into a lot of detail, but we said that each of them serves as sort of biological information, genetic information. Unlike a cell that has both of these types of molecules, any one virus will either have this or that, but not both. And in fact, one way to categorize or classify viruses is basing the classification on whether or not they're a DNA virus or an RNA virus. Some have just this, some have just that. And how they behave in the host is going to depend on which they are. So that's another feature to know about. Some, not all, certainly, in fact not most, but some viruses are oncogenic. And I'm going to spend a few minutes telling you about what that means a little bit later on, a couple pages from now. That onco means cancer. So in other words, some viruses are related to some forms of cancer, and we're going to talk about why that's the case as well. Let's leave a couple of these open, and then we can go back and fill them later. But these are just a couple of general fun facts. Actually, let's fill in one more. Some viruses, not all, have a structure called an envelope, a viral envelope. And we'll mention that on the next page too when we talk about how viruses work. So again, these are very, very simple. They're way smaller than even the smallest bacterial cell. It's just a little protein shell, piece of nucleic acid inside, and maybe or maybe not this other structure called an envelope. And I'll tell you about that on the next page. So if you flip to page 108, you're going to see a place to write some steps. What we're going to talk about here, whether we're talking about a virus with an envelope or without an envelope, there are just a couple of basic steps that are going to occur when that virus infects its host cell. So let's say that that's a host cell. And let's say that we have a virus show up. Let's start out with a non-enveloped virus first, a virus that does not have a viral envelope. It's just a little protein shell called a capsid. I'll just draw it like that. There's a capsid. And there's a little bit of nucleic acid inside. I'll just draw a little squiggle inside for the nucleic acid. Meanwhile, this cell is much larger than that virus. So this virus shows up. What is that cell going to do? Well, that cell in all likelihood doesn't even recognize or realize that that thing is there. The virus, on the other hand, wants to get into the guts of that cell. Out here, it can't do anything. In there, it can hijack the machinery of the cell and convince the cell to make copies of itself. And that's really the goal of the virus, is make copies of itself. It just doesn't have the capacity to do it on its own. So it's got to get from here to there. Think back to membrane transport. That was a recent topic a few weeks ago. There's videos up about it. You answered questions on exam number 4 about it. Remember active transport, and remember specifically endocytosis. There was a step-by-step process that we talked about back then. Well, what if this cell endocytized that virus? Well, that virus would wind up inside the cell then. And you might say, well, why would the cell want to do that? This thing is going to cause it harm. Why would it be inviting the virus in? Well, the virus sort of fools the cell into thinking that it should endocytize it. You see, on the outside of cells, potential host cells, are these proteins called viral receptors. So there's a viral receptor. I've drawn one there. Imagine there's lots more out there. These are proteins. They're worn on the surface of the potential host cell, and they're there for completely different reasons than what we're going to talk about. In other words, the cell would have built that protein because it needed it for something. It needed it for its own normal activity, for its own life. The virus recognizes this. The virus has sort of co-evolved with the host to be able to say, oh, that cell wears these receptors. I'm going to design a mechanism. I'm going to design myself so that I can attach to that particular molecule. And it will. It'll bind. It'll fit. It's almost like a little lock and key. This virus capsid recognizes that viral receptor. That molecule that is on the surface of this cell is receptive to this virus. It will be able to attach to this virus. So when this attaches here, that will induce this cell to endocytize the virus. Remember receptor-mediated endocytosis from back when we discussed active transport? If not, please go back and look up the video in that section, the cell membrane transport section. There was a little blurb in there about endocytosis and then about receptor-mediated endocytosis. And this is exactly that. So the virus is finding a way to get into the host. The host cell is saying, oh, I have this protein here, and it's got a normal function, has nothing to do with viruses, but somehow this virus learned how to attach to that protein of mine. And when it does, I'm going to unwittingly endocytize this virus. And without drawing all the steps of endocytosis, I'm going to say that eventually that results in this virus getting to the inside of that cell, which is exactly what it should have done. So what we just looked at here is attachment and entrance. The attachment to the host cell is very specific. It's based on the presence of a receptor being there. And then the entrance has to do with endocytosis. In this case, specifically receptor-mediated endocytosis. You get the virus into the guts of the cell. So again, the host cell didn't realize what it was doing, if you want to think of it that way. It invited this virus in because it thought it was supposed to. But now, eventually, of course, it's going to realize, gee, that was a bad idea, because now I have a parasite inside of me. So the virus is sort of developed the capacity to trick the host, if you will. So that's attachment and entrance. There's something really important to note about this. Let's say this is one particular cell type in your body. I'll make up an example. Let's say that this is a liver cell. And let's say that's a hepatitis virus. Now, hepatitis viruses only infect liver cells. Here, I'll write that down. Hepatitis. Hepat means liver. So hepatitis viruses only infect liver cells. Why don't they infect skin cells or kidney cells or bone cells or muscle cells? Because those other kinds of cells don't have the receptor that a hepatitis virus will fit into. Liver cells, on the other hand, do. So hepatitis viruses can infect liver cells. In other words, the presence or absence of the viral receptor for a particular kind of virus will indicate whether or not this cell can be a potential host for that particular kind of virus. And that brings up a really important point that you're going to need to know about when you discuss viruses. It's the idea of what's called host range. When you talk about the host range of a parasite, or in this case specifically a virus, you're talking about the breadth of hosts that can possibly be infected. In other words, how many different kinds of cells can a particular virus infect? Some viruses have a very broad host range. You may have a broad host range, and that means they can infect a lot of different kinds of cells because a lot of different kinds of cells have the right receptor. Other viruses, on the other hand, have a very narrow host range because there's only very specific certain kinds of cells, or very few kinds of cells, that have the right viral receptor. A couple examples. A rabies virus. That will infect nerve cells of mammals. Now that's pretty broad. There's a lot of different kinds of mammals, and their nerve cells, because they have the right viral receptors, are potential hosts for rabies viruses. A hepatitis B virus, for example, we'll say this is hepatitis B. There's different kinds of hepatitis. We'll say it's a hepatitis B virus. That can only affect liver cells of primates. So only certain kinds of mammals and only their liver cells can be infected. That's a more narrow host range. So infectious disease specialists will use these terms all the time, and will talk about how broad or narrow a host range is. And for viruses, that's to a large extent dictated by the presence or absence of the viral receptor on a particular kind of host cell. Okay, so we've got attachment and entrance, in this case by endocytosis. What happens once that virus gets into the host cell? Well, once it's in there, here's that host cell again, it will get disassembled because the capsid has served its purpose. The capsid was just a little shell for the nucleic acid to be carried around in. Once that capsid is disassembled, you get that little piece of genetic information there, that little piece of DNA or RNA. And without going into all the hardcore details on this, that piece of DNA or RNA is now going to serve as instructions on how to make more viruses, how to synthesize more capsid proteins, how to assemble them into new capsids, how to make copies of the nucleic acid itself. So that's the instructions on how to make more viruses. And the host cell is really good at reading genetic information. It does it all the time. It does it to keep itself alive. It knows how to handle a piece of DNA or RNA. It, in this case, kind of doesn't notice that this little piece of nucleic acid is not its own. It just says, hey, here's a piece of information. I'm going to read it, and I'm going to follow the instructions that I find. But it turns out that the instructions here are going to be instructions on how to make more viruses. So you get this event called replication and assembly. In other words, you get lots and lots and lots of little capsids being built in here, and you get lots and lots and lots of extra copies of that viral nucleic acid being made. And before you know it, you have hundreds of thousands or millions of copies of what was just one original virus sitting inside that host cell. In other words, the virus couldn't copy itself, but it can get into the host cell and convince the host cell to make copies of it. And of course, this is to the detriment of the host cell. It's using its own resources, its own raw materials, its own energy to build viruses, which don't serve any useful purpose to it. Once those viruses have been assembled, and again, there's maybe hundreds of thousands, maybe even more viruses packed into that cell. Once that's happened, it's time for those viruses to leave the cell and go infect other cells. And so that's the process of release. So how do these get out of the host cell? Often it's just that the host cell simply ruptures. There's so many of these packed in there that the cell just breaks open. That's just a process called lysis. And the cell obviously is going to be dead as a result of that, but all these viruses can then go infect neighboring cells and do the same thing to them. So one cell dies and then a whole bunch more and so on. In some cases, release from a cell is a little more organized. In some cases, if you think back to active transport, exocytosis of these will happen. So the host cell will remain alive, but it will sort of in an organized way extrude these viruses, and they'll still wind up getting out, but not in such a crude way as just hacking the cell open. There are also times where the virus brings with it information on how to make an enzyme that will cut the cell membrane open. So it may be pressure as a result of so many viruses being packed in here. It may be an intentional cutting open of the cell membrane that allows these viruses out, or it may be that the cell is induced to exocytize those viruses. No matter which method happened, release is what we've just been talking about, the exit of the viruses from the host. Now, how is all of that different if there's an envelope on this virus? Well, the overall steps are going to be the same, but how they occur may be slightly different. So just a couple quick words on what a viral envelope is in the first place. We've mentioned it already. All viruses are going to have a capsid, this little protein shell. They're all going to have the little glob of nucleic acid inside, but some of them will also carry around this extra little feature, I'll draw it in blue, called a viral envelope. Again, we mentioned that on the previous page. And if you look at the molecular makeup of the viral envelope, it actually looks like cell membrane. Now remember, this is not a cell, but it's made mostly of phospholipids. Remember the phospholipid bilayer and the cell membrane? It looks like that. And there's some proteins in there as well. In other words, kind of looks like a little miniature cell membrane. Well, those proteins that are built into that viral envelope include what are called spike proteins, these little protruding proteins like this. And those little spike proteins, in the case of an envelope virus, can help that virus find a receptor and stick to a receptor. So they're going to aid in the attachment of this virus to its potential host cell. Some viruses simply don't have an envelope and just do what we said before. Others have this extra little feature. It all depends on which kind of virus you're talking about. So that's a quick run through. Of course, each of those steps is way more complex than what I've implied in just this short video, but that's the overall picture of how a virus finds its host, selects one cell versus another to be a host, gets in there, gets copies of itself made, and then gets released so it can go find more hosts. And I'm going to stop the video here, and I'll make another one to tell you specifically about things that are on page 109, and that's oncogenic viruses.